You can start now.
Hello, everybody. I will be talking about the coronavirus, its involvement in neuroinflammation and potentially in brain fog. We all know that there are many neurological symptoms associated with long COVID. I chose to show some slides here to make the point, but probably most importantly is the publication shown at the bottom where they talk about post-COVID symptoms are associated with endotypes reflecting poor inflammatory and hemostatic modulation. That leads us to what we will be talking about, that is inflammation and potentially perivascular or endothelial associated inflammation. There have been a number of suggested pathogenetic processes that may be involved in long COVID. In the color box, I indicate blood-brain barrier disruption. And on the right-hand side, you can see a number of recent papers talking about alterations in BBB integrity in COVID patients. There is perivascular inflammation. And on the left-hand side, you see how the spike protein can impair endothelial function, as well as a paper showing that in the brain of patients affected, there is actually microvascular injury. There's also microglial activation, and at the bottom right, you see at least one paper talking about microglia activation in patients with COVID, with and without dementia. And finally, neuronal damage. And on the left-hand side, you see mild respiratory COVID being associated with neural damage. At the bottom, you see brain injury in COVID-19 patients associated again with dysregulated innate and adaptive immune responses, but probably most importantly, the Nature paper shown in the center, where they showed that there is actually long-standing neurodegeneration associated with microglia activation in the brain. So I don't think there is much doubt that there is inflammation in the brain without necessarily infection of the brain by the live virus. So we made the hypothesis that a unique mast cell, an immune cell found everywhere in the body, including the brain, may be involved. Why? First of all, its location is perivascular. And as you can see at the upper right-hand side, you see a cross-section of a blood vessel, and all the red arrows indicate those violet cells. These are all mast cells. Of course, we know the mast cells because of their involvement in allergic reactions, but we know much less about what they do in the brain. They do regulate blood-brain barrier permeability. I wrote a review back in 1990 where I called the mast cells the immune gate to the brain. And at the bottom left, you can see a caricature of the blood-brain barrier with the lumen, endothelial cells, pericytes, and at the top, a little mast cell. But if you look at the center lower panel, you see an electron micrograph from our laboratory, magnified 300,000 diameters, where you see a red blood cell in the middle of the lumen. You see endothelial cells marked with an E, P is the pericyte, and in 11 o'clock, you see actually a mast cell hugging the blood-brain barrier. So the mast cells are found not only perivascular in the brain, they're found in the meninges, as you can see at the bottom publication. And very importantly, they're found actually in areas that regulate homeostasis and behavior. However, they do not actually live in isolation. The mast cells do communicate with the microglia, which of course are much more plentiful in the brain than the mast cells. And you can see papers where the mast cells are involved in neuroinflammation alone or with microglia. And very importantly, at the bottom, you see two publications where mediators released from the mast cells at the left-hand side, and especially this proteolytic enzyme tryptase on the right-hand side can in fact stimulate microglia, which we all know are the defenders of the brain, and they're involved in inflammation in the brain. Now, what do we know about mast cells? So this is basically a quick review of a mast cell. At the 11 o'clock site, you see the receptors for immunoglobulin E. Those are the receptors that engage antigen, and that quickly leads to what we call degranulation, where the granules release their content to the outside, as you can see at the bottom left, where the intact granules are dense blue, and those that are releasing the content are actually swollen and they're light violet. And within minutes, they release numerous molecules such as histamine, heparin, proteolytic enzymes like chymase, and tryptase, platelet activating factor, which is involved in clotting, and tumor necrosis factor. And in fact, we wrote a review in the New England Journal of Medicine about mast cells, mastocytosis, and related disorders. Now, in addition, over the last few years, there was a new receptor discovered, MRGPRX2. And what is important is this receptor is stimulated by positively charged molecules, whether it's neuropeptides or drugs. And as you see at the publication on the left-hand side, this receptor communicates with the Ig receptor. And it seems like when one is activated, resets the reactivity of the other receptor. So the mast cells now are ready to fire much more. But at the right-hand side, I indicate receptors such as for neuropeptides through high affinity receptors like neurotensin substance P, for cytokines like interleukin-1 and interleukin-33, and toll-like receptors for bacteria, fungi, and viruses. And it's very important to keep in mind, as the caricature shows from the New England Journal of Medicine article, that the mast cell shown at the center can be triggered by numerous other triggers than Ig and antigen. So the mast cell can really respond to pretty much everything and that orchestrates an inflammatory response. That response, through the activation of the receptors at the right-hand side, occurs without degranulation. And as you can see at the bottom rectangle, cytokines, chemokines, and pro-inflammatory peptides are being released. Very critically, we published 2 papers in the Proceedings of the National Academy of Sciences showing that if the mast cells are stimulated by neuropeptide substance P, and this alarmin, the cytokine interleukin-33, they put out thousands of units of TNF, tumor necrosis factor, and in the middle panel, hundreds of units of interleukin-1 beta. In fact, these amounts of TNF and IL-1 beta are more than any other immune cell that I've actually been studying. And to make things worse, other colleagues show at the bottom publication that once the mast cells are activated, they release a molecule called hemokinin-1 that's very similar to substance P, which now acts back in an autocrine fashion and further stimulates the mast cells. So because of all of these findings so far, we made the assumption that the mast cells, especially in the lungs, may be involved in COVID. Why? Well, we know the mast cells are found in the lungs because of their involvement in asthma. They can release cytokines, they can release platelet activating factor, so they can cause edema, inflammation, and macroemboli, which are the telltale sign, basically, of what we have seen in patients who died with COVID. And at the left-hand side, I indicated some papers which are association studies reviewing the evidence that the mast cells may be involved in COVID. But in the right-hand side, there's specific papers proving the point. For instance, mast cells release platelet activating factor. And in fact, we published at the upper top with colleagues from Greece that the coronavirus can stimulate endothelial cells to produce actually PAF. The middle panel shows 2 papers where mast cells were found to be increased and degranulated in the lungs of patients who had died with COVID. And at the very bottom, uniquely interesting, histamine, which of course comes only from the mast cells, was shown to potentiate the spike protein entry into endothelial cells. Recently, there have been additional proof that mast cells are involved in COVID. At the left-hand side, colleagues infected mice with the coronavirus, and they showed that at the top blue, the lungs had normal-looking mast cells, but the infected mice at the bottom showed degranulated mast cells indicated by the red arrows. Then they measured chymase in the blood of these mice, and as you can see at the top graph, over a few days, there was a lot of chymase being released. When they looked actually at patients with different degrees of severity, they showed that the more severe the symptoms, the more chymase was being released. In another paper showing at the right upper hand side, again, colleagues measured either chymase or tryptase, these unique mast cell related enzymes, and they showed as compared to healthy controls, in both cases, chymase and tryptase were increased. And I want to stress with the paper shown at the right hand side in the middle, that mast cells can promote viral entry of coronavirus by formation of chymase spike protein complexes. The paper at the bottom shows that when human culture mast cells were used, and they were stimulated either with a synthetic compound called C4880 or the receptor binding domain or the virus. There was a lot of tryptase and chymase released from these human cells, but I have to state that the amount of RBD they used was actually pharmacological, much more than one would have expected. We were though intrigued by the fact that interleukin-33 seems to be affecting the release of the mast cells, and we had written a publication, a review about interleukin-3 in autoimmunity and inflammation. So about a year ago, we published with colleagues from Greece a paper where we studied pro and anti-inflammatory cytokines, and uniquely we showed that the soluble receptor for interleukin-33, showing at the left bottom, was actually increased as the severity of the patients increased. So you see severe patients, and then at the very right, intubated patients, and this soluble receptor for interleukin-33 was much higher. Well, that was an oxymoron because we know interleukin-33 is involved in inflammation. So our take was that the body was trying to contain the inflammation by releasing soluble receptors that would be neutralizing interleukin-33, but obviously that wasn't sufficient to stop the progression of the disease. So what we did, and we published this as a letter to the editor in the New England Journal of Medicine back in May, we actually used recombinant spike protein as compared to substance P using human culture mast cells, and we measured release of interleukin-1 beta, and we did that actually in the presence or absence of interleukin-33. And as you can see, interleukin-33 augmented the release of interleukin-1 beta from human mast cells. So there's no question that the mast cells can be activated by the coronavirus, and the presence of interleukin-33, which is released from dying or damaged cells, will actually worsen the situation. Then we published recently a more comprehensive paper where we looked at the release of various different molecules under different conditions. Let me start at the left-hand side. So again, we use substance P as a control. We used with or without interleukin-3, full-length spike protein, or the receptor binding domain. And we measured at the top A and B panels, chymase and tryptase. And as you can see, of course, the whole length protein stimulated release of both proteases, but the receptor binding domain also had an effect as well. You'll see why this is important, because when we move to the right-hand side graph, you see we use now antibodies against either the toll-like receptor 2 or toll-like receptor 4 or the ACE2 receptor, and it was only the ACE2 receptor that blocked the response actually to the RBD, but not to the full-length protein. When we looked actually at interleukin-1 beta release on the C panel at the right-hand side, it was only TLR4 antibody that blocked the release of that. And you go to the left-hand side where we compared the full-length spike protein against the RBD. Again, the RBD was very weak in inducing release of interleukin-1 beta. That indicates that, at least in the mast cells, the spike protein can stimulate different receptors and lead to the release of different pro-inflammatory molecules. And it is not only the ACE2 receptor that is involved. How about brain fog? How can we connect the dots? Well, brain fog is a slang term, but it can be described as mental cloudiness, mental fatigue, difficulty focusing, difficulty finding the right words, multitasking, as you can see on the cover at the left-hand side. There are many papers indicating there is cognitive impairment or mental fatigue, if you wish, in COVID-19 patients. But I want to stress that this is not unique to just long COVID patients. At the left-hand side, you see evidence of cognitive impairment in mastocytosis patients who have many more mast cells that are also activated. At the left-hand side bottom, you see brain fog in chronic fatigue syndrome or myalgic encephalomyelitis chronic fatigue syndrome. And many publications that brain fog is a key symptom in chemotherapy or patients in undertaking chemotherapy, and many people call that chemo fog or chemo brain. And I dare say that I believe that the underlying mechanisms that I'm describing today might in fact be similar in all of these instances. So we wrote a review about a year ago or so, asking whether the spike protein might be responsible for long COVID syndrome. But then someone might ask, well, okay, mast cells may be activated in the lungs in the acute phase of the infection. Well, how about in the long COVID? Is the spike protein still sitting around being available to activate the mast cells and the microglial cells? And in fact, there are two very good papers proving that point. You see the right upper hand side, persistent circulating severe acute respiratory syndrome coronavirus spike is associated with post-acute symptoms. And a paper in the middle panel showing persistent circulating spike is associated again with sequelae. And in fact, there are other publications that when brain mast cells are activated, they can lead to cognitive dysfunction under different circumstances, and even food allergy can induce brain inflammation and cognitive impairments, and of course, allergies associated with mast cells. So if you see at the caricature at the left-hand side at the bottom, we're showing the blood-brain barrier. The mast cells regulate the blood-brain barrier. If they're activated, they will make it leaky. The spike protein or the whole coronavirus can enter, then damage the endothelial cells, activate the mast cells can activate the microglial cells, and you end up with a localized inflammatory response. What is the evidence that the mast cells regulate the blood-brain barrier? We showed that with many publications. At the top, we injected technetium gluceptate into mice. We stressed the mice, and as you can see at the black bar at the right-hand side, most of the technetium gluceptate went into the brain parenchyma. When we did that in mast cell deficient WW mice, nothing happened. Then we showed by using antibodies against the CRH R1 receptor that the mast cells express, these are human mast cells, express the receptor as shown by fluorescent antibody against the receptor. And when they're stimulated, as you see at the graph at the right-hand side, they put out a lot of vascular endothelia growth factor, the upgoing curve, while there's actually no tryptase release. So this is unique selective release of vascular endothelia growth factor, which is also vasodilatory. And then we also went back to interleukin 33, published in PNAS, and we showed that interleukin 33 augments substance P-induced VEGF release. And to make things more complicated, the publication at the bottom shows that substance P stimulates the mast cells to express receptors for CRH and vice versa. So obviously the mast cells are very dynamic cells responding to whatever is released in the environment. So then we turn to the microglial cells, and we published this recently, and we used again coronavirus spike protein or receptor binding domain. And as you can see, we measured interleukin 1 beta first. You see the spike protein full length could stimulate release of interleukin 1 beta, but not the RBD. Same thing with IL-8. The full length protein stimulated, but not the RBD. But when we went to TNF and protein S100 beta, which indicates neuronal damage, you see that both the full length spike protein and the RBD could stimulate release. Again, different receptors seem to be stimulating release of different molecules for the microglia as well. And we use the same paradigm we use for mast cells. We pre-incubated the cells with antibodies against either TLR2, TLR4, or the ACE2 receptor, showing at the left bottom. And the red rectangles indicate that only TLR4 antibody reduced the release of interleukin-1 beta and interleukin 8 and had no effect on TNF alpha and S100 beta. And at the bottom, you see at least one of a number of papers where other colleagues have shown that, in fact, TLR4 is activated. Not only is activated, as I've already shown you, but in this paper, it shows that TLR4 activation that leads to further increase of the ACE2 receptor, which further complicates things because we have a self-supporting paradigm. Well, what can we do about any of this? We have been studying natural molecules called flavonoids for almost 30 years, and we focused over the last few years on a molecule called luteolin, which is anti-inflammatory, antioxidant, blocks mast cells, blocks microglia, as you can see in the right upper hand side box. And a number of publications, some from us, some from others, have shown that luteolin is not only anti-inflammatory, but it's also neuroprotective. And in fact, at the bottom right, you see a publication of ours where we reviewed the evidence that luteolin can actually inhibit or prevent brain fog associated with long COVID syndrome. We actually showed that the luteolin and a similar molecule called methoxyluteolin that has methyl groups instead of hydroxy groups can block release of mast cell mediators, showing at the right hand side, where we compare it to the drug cromolyn, supposed to be a mast cell inhibitor. Luteolin was better, methoxyluteolin was even better. We did this with microglia as well, stimulated with neurotensin, and as we increase the pre-incubation amount of these flavonoids, you see both luteolin and methoxyluteolin inhibited release of interleukin-1 beta from human culture microglia almost 100%. And very importantly, a paper published very recently at the bottom where it shows the luteolin inhibits actually spike protein binding to the ACE2 receptor. So you can see how molecules such as luteolin might affect positively the outcomes by inhibiting binding to the ACE2 receptor and inhibiting the effect of the TLR4 activation. But these flavonoids are lipid soluble. So we were looking for a flavonoid that might be partially at least water soluble. And we discovered that eriodictyol, which is very similar to luteolin, almost identical, except it's got a planar, basically benzene ring, can in fact do that. And we compared at the right, at the bottom left, the ability of luteolin or methoxyluteolin or nobiletin, which is actually tetramethylated, basically luteolin, versus eriodictyol for the ability to block lipopolysaccharide-induced interleukin-1 release from human culture microglia. And the reason why we chose LPS, because LPS actually works by activating the TLR receptor, and it's a very strong, very potent activator of the receptor. And you can see all these flavonoids can inhibit, and a 50 micromolar eriodictyol was the best. And at least 2 papers have shown that, in fact, eriodictyol is unique. The paper shown in the middle, they screen about 1.2 billion small molecules for their ability to block binding of the coronavirus to its receptor, and eriodictyol was among the top 3. And at the bottom, there's another recent paper showing that eriodictyol suppressed the cellular entry again of the coronavirus. So in addition to what it does vis-a-vis inflammatory mediator release, at least eriodictyol, as some other flavonoids, can block the ability of the coronavirus to enter the cells. And the caricature at the right upper hand side basically shows again how toxins and viruses can enter the brain, especially in the area of the amygdala and the hypothalamus that regulate behavior and homeostasis, and how these triggers can either trigger the mast cells to then trigger the microglia to then release molecules that might lead to neuro-COVID, or they might stimulate the microglia directly. But in either instance, eriodictyol seems to be inhibiting both the mast cell and the microglia activation. The final slide, which is a little more complicated, is here. Let me start with a summary of the take-home messages. So long COVID involves perivascular inflammation. I think that's a given. The coronavirus, especially the spike protein, can activate mast cells and microglia. I think I gave you enough evidence for that. Mast cell is known to disrupt the blood-brain barrier and orchestrate an inflammatory response. However, there are no effective drugs to block mast cells or microglia, or of course prevent or treat neuro-COVID or long COVID. So I recently developed a natural dietary supplement in vegetarian capsules that is called Viral Protect from a company that is actually registered by the FDA. And if you export these molecules, the FDA can give you a certificate of free sale shown on the right-hand side. So what we decided to do is combine 4 molecules. So we combined eriodictyol with luteolin. I've already given you the evidence for that. So those will block the TLR4 receptor, the ACE2 receptor, and the serine protease, which is important to cleave the spike protein and allow the virus to enter the nucleus. We added sulforaphane, and at the right-hand side in the middle, you see one publication where sulforaphane actually can be used because it's got antiviral activity against the coronavirus. We added oleuropein from olive leaf extract. And in the middle panel, I'm showing at least a paper where alluropoint through a computational biology approach was shown to be at the very top of molecules that can actually block the coronavirus, especially the neurologic complications of the virus. And at the bottom, it's a case report that I published where we used actually these products to help a very severe patient with COVID and neuro-COVID as well. So in the middle panel, I'm showing basically a diagram, which I was referring to, how we can basically address various targets. And as you can see, the iridicyclic and the luteolin can block the receptors and the serine protease, while oleuropein, hydroxytyrosol, also from olive leaf extract and sulforaphane, can block the inflammasome, the serine proteases, as well as the multiplication of the virus. And as we know from pharmacology, the more targets we actually block, the more likely that we will have a real beneficial effect. For whatever it's worth, if you look at the bottom left, I've been awarded a US patent for use of flavonoids for brain inflammation. And I have actually submitted a patent application, which is pending, about the use of very addictive compositions and methods, focusing, of course, in long COVID. So as a summary, I believe that there's a lot of evidence of molecular events that associate the coronavirus, especially spike protein, with neuroinflammation, perivascular inflammation involving mast cells and microglia. By using those as targets, clearly we can at least identify molecules that might be beneficial and combine the molecules. Now you might ask, so far so good, but how can we actually show that in some kind of a surrogate model for long COVID? Well, as we all know, there's no model for long COVID right now, but what we're actually doing right now in the laboratory is to use a brain on a chip approach with organoids. So we're using basically a center panel where we can put neurons, and with microfluidics, we can put actually different cell types in satellite wells. So we're using mast cells, microglia, and endothelial cells. We can stimulate each culture separately or the cultures together, measure the activity basically of the neurons and the health of the neurons in the middle. And we're using this surrogate model to basically screen various natural compounds or drugs for their ability to either block each one of the various cell types or prevent the damage to neurons. We believe that this brain on a chip approach is probably the best way to try to mimic a complex neurologic disease moving forward. Thank you very much for listening.